All right, so hi everyone, I'm Gillian Rowan. I also work with Margaret at McGill University, and on a slightly different note, my work focuses on applying remote sensing to aquatic vegetation and aquatic environments. Deep, you can go to the next slide. So to start, why am I focusing on aquatic vegetation in the inlands? Well, most of remote sensing thus far has been conducted on coastal environments, where you have type 1, type 1A, type 1B, or type 2 waters here. So you can see that there would be 30% of the light penetrating down to 10.5 meters into the water column. Whereas in an inland or freshwater ecosystem, getting that 3.7, which is considered the worst water quality for coastal waters, would be very, very good and very exciting. So a lot of the work has focused on where the visibility is best. Additionally, the inland waters have all this aquatic vegetation that's extremely beneficial to the ecosystems and to us because it improves water quality, it stabilizes sediments, and also creates habitat. So basically, there's a really big and very important knowledge gap that I would like to fill. Next slide. So this is a little bit more of a graphical illustration of that knowledge gap. I looked at every past study I could find, and put together what sensor they used for what application. And the colored boxes show the accuracy of the results that they obtained, whereas all of the gray boxes show work that I haven't found any indication of having ever been done. So there's clearly a huge gap and a need for more research into this, this application of remote sensing. Next slide. So I found that there were these 5 main targets that kind of defined whether aquatic remote sensing would be successful, and it's target separability. So what are you looking at and how are you looking at it? Water depth and water type, which are functions of the medium itself. So how thick is that water column and how does it interact with the light? And then spectral resolution and spatial resolution are factors are determined by your imaging methods. So which sensor are you using, which platform are you using? And if we look at, for example, the panel A here is a microcassi image where you can see down to the plant level versus a cassi image on the right, panel B, would leave you limited to a canopy level information. Next slide. So we're going to start off with target separability. Next. Okay, to look at target separability, I collected 85 samples of vegetation in the summers of 2019 and 2020 from the Longso Parkway field site. I had 13 species in total that covered 11 genera. For example, this middle plant that's shown here is very interesting for my work because it's an invasive species, Myrophyllum spicatum. So ecosystem managers are very interested at being able to track where it is. These plants were collected, brought to a lab, were measured, cleaned, measured again, and then pressed for a record. And I measured them twice so that I could get spectral profiles of them as they were with and without epiphyte fouling. Next. So here I have the average spectra for each of the 13 species shown. And clearly they follow a similar shape to the vegetation that we're used to, but the reflectance is significantly lower in magnitude. I then used all of the spectra that I collected to perform a dimension reduction through feature selection and to determine which wavelengths were important to classifying the samples. Next. So through this feature selection, I found significantly smaller data sets or usable features here. I originally included about 1,100 data features and got it down to a couple hundred or as low as 57 here. And you can see that all of the 2019 data sets had fairly good maximum separability, but the 2020 data set had significantly less. So next slide. This can be determined or can be attributed to the greater variation that there was in the 2020 plants. So on the left side, we can see something that's representative of what was harvested in 2019. And on the right, we see something representative of 2020, where there's a lot more variation. It goes from green to red. And this variation within a species, so intraspecific, makes the classification amongst the species much harder. And that led to the reduced separability. Next slide. So once the feature selection was completed, I performed a classification to distinguish between the 13 species. And you can see I got some very promising results. Between 77 and 81% between 13 species is very, very high classification accuracy. If, for example, though, you weren't interested in grouping by species, if you grouped by genera, it's a little bit higher, even at 80%. Or if you're trying to distinguish something between like plants versus algae, so by the kingdom, you have exceptionally high classification accuracy of 95%. Next slide. So that's my first look at target separability, and now we'll move on to the water depth and water type. Next. So to do this, I used the spectral panel as a target because it was the brightest possible option and it reflects light consistently. So I put the spectral panel underwater and I measured it at 5 centimeter intervals from 0 to 115 centimeters in depth. And I did 3 transects like that per location. And I did that for 2 locations, one in a river and one in a lake. Next slide. So here's an example of what I got for the lake location. And ooh, I think you skipped a slide there, Deep. Yep, there we go. So here depth is shown as a variation in the shade of the line, the lightest blue being 0 centimeters. It's up a little bit above 1. And then the darkest being 115. And clearly there's very little signal above 950 nanometers for any depth. So from here on out, I'm just going to focus on the lower end of the spectrum. Next. There we go. So here I have both curves side by side for the 2 locations. And you can see that the reflectance profiles take on pretty similar shapes and magnitudes, but they're not identical. And specifically in the visible portion of the spectrum, it gets a little messier. So let's dig into that a little further and we can go to the next slide. So to look at that a little bit closer, I picked out 6 wavelengths of interest. Here shown in red are 370, 750, and 810. And I chose those because they show a consistent relationship to depth. So with deeper depths, there's weaker reflectance. And then 450, 565, and 680 were shown because they don't follow that relationship. Like there are a few areas where reflectance actually increases with depth. And I wanted to take a look at why that was happening and what was going on there. Next. So I plotted each of these 6 wavelengths against depth, and I found that for the 3 that followed that relationship, 370, 750, and 810, they're all dominated by processes attributable to the water itself, so the scattering and the absorption of water, and they show uniform exponential decay according to depth. And then if you look at the 450, 565, and 680 wavelengths, they're in the region of the spectrum that's dominated by the water column constituents, so pigments, tannins, things like that. And those are the aspects that really vary between water types. And these wavelengths present differently shaped relationships according to depth. Next. So now if we switch to look at the other water type, the river, we can see the very strong relationships in those 3 wavelengths that were dominated by the physical processes. They have a similar shape and magnitude to that other water type. There's very little change in those wavelengths, but the behavior in the visible region is markedly different. Next. There we go. So if we go back to the curves, you can see that very similar behavior between the 2 locations where the physical processes dominate. So if you wanted to translate the results between 2 different water types, it seems like you would focus on the portions of the spectrum where the water column has that similar effect. So in these smaller wavelengths and the near infrared region that's still visible. But within one water type, if you wanted to translate results between different depths, you would want to focus on the visible portion of the region where the water column attenuation is the least strong, so you get that maximum penetration or penetration to a deeper depth. Next. So to combine the two portions of my work so far, I've plotted the features selected for one of the 2019 data sets over the water reflectance curves. So the features are graphed according to rank, with the lowest rank being the most important features and the higher ones contributing less to the spectral separability. And as we can see, there aren't many important features above that 950 mark, which is great because in situ situations, we wouldn't be able to see those. Next. And if we zoom in a little bit closer on that portion that is usable, we can see that they're even clustered within the spectral portion or the visible portion of the spectrum. And interestingly, it looks like they're also somewhat grouped around 3 vertical lines representing different wavelengths. Next. So here I've put those lines that I kind of saw as groupings, and they are at 550, 680, and 445. 550 and 680 being absorbance and reflectance peaks of chlorophylls itself. And then 445 is a shared absorbance peak of all the major leaf pigments. So this is just a rough illustration showing that leaf pigments are the major drivers in submerged aquatic vegetation spectral separability. Next. So then I took those 2 different portions of the spectrum that are usable for, or are useful for different applications. And I cropped the features selected for the data sets down to those regions. And then I reclassified them and examined how the classification accuracy was affected. So we see that the classification accuracy is reduced when the portion of the spectrum being examined is reduced. Makes sense. But using either the UV, vis, and near infrared, which would be the different water types one, or the visible one, which is for different depths, you still get pretty reasonable classification accuracies between 50 and 70%, which again, this is trying to distinguish between 13 different species, which is a pretty complex classification problem. And then if, same as before, if you weren't interested in species, you wanted to look at something like the genera or the kingdom, you again have that higher classification accuracy with separating between plants and algae being quite reliable under every condition. So it could be potentially useful for different water types and different depths. Next. So here to summarize, in terms of target separability, the classification by species is definitely possible and accuracy is improved with coarser groupings. The water depth can be addressed by focusing on the visible portion of the spectrum. Water type can be addressed by focusing on that extremities of the visible portion of the spectrum. And I still have work to come on how spectral and spatial resolutions affect the success of aquatic remote sensing. Thank you all for your time and attention, and I'd love to answer whatever questions you have.
Thank you very much, Gillian. So we have time for one short question. I see no hands, no one in the chat.
I have a question, so Gillian.
Yeah, go on.
So obviously water depth is very important. So I don't know much, it's a beginner question, but you know, we can use LiDAR pretty well to estimate water depth. So it'd be really important, I guess, if you want to know the water depth. So what sort of remote sensing methods can give you accurate measures of water depth?
Can you repeat that?
Well, I guess, you know, obviously the depth at which the plant is will really strongly affect the reflected signal, right?
Mm-hmm.
So, and so if you wanted to remove the water, I guess you do need to know what the depth is. So I was just curious, what does, you know, how accurately can LiDAR estimate the water depth of particular plants, for example?
So I wouldn't say LiDAR is necessarily the most used. There is bathymetric LiDAR, which can tell you the bathymetry, but the plant height or the thickness of the water column above plants obviously varies depending on how tall the plant is. There are different equations and models that are used to estimate the thickness of the water column, but those are all developed for particular sensors or particular water types. So they're not necessarily transferable. I'm going to be looking a little bit more at how well you can determine the water type and the water column thickness. But there isn't necessarily one like reliable cure-all for that problem.
Thanks.